Hi, I am Dilip. This video discusses about the project CRISPR as a cure to HIV which I have done in SC462. Now, we will first discuss about the technology tool which opened a path to find cure for many diseases. CRISPR, known as CRISPR-Cas9. What is CRISPR-Cas9? CRISPR-Cas9 is a genome editing tool which evolved from tools such as zinc finger nuclei or transcriptor activator like effector TALEN or the ZFN. CRISPR-Cas9 can easily identify a specific site of nucleotide sequence of targeted genome sequence. This system is originally came from the adaptive immune system of the bacteria and archaea. Now, what is the role of Cas9 in it? Cas9 acts as a scissor guided by gRNA to cut at the specific site matching gRNA sequence. Now, most CRISPR-Cas9 is nowadays a very popular Cas9 genome editing tool. Now, most useful application of CRISPR-Cas9 is seen in the transgenetic mutation. There are many written applications on this slide as you can read. But the most useful application of CRISPR-Cas9 is the transgenetic mutation. It also helps in curing many diseases. Now, as a project talks about the CRISPR as a cure to the HIV, so we will first understand how does HIV work. Now, to understand, we need to look at this working process. HIV infects the host in 4 stages. The 4 stages are as written: binding and fusion, reverse transcription and integration, transcription and translation, assembly binding and maturation. Now, to understand these stages, we will talk about them a little bit. As the details are written in this slide, but we will try to understand from the figure given in the next slide. Now, in the next slide, as it is written, the first stage is the binding and fusion. Now, HIV virus starts by attaching itself to the T helper cell. HIV particle contain spikes which help them to join with the cell. Then starts the transfer of contents of HIV particle into the cell. The second stage is the reverse transcription and integration. Now, HIV is inside the body. It starts translating HIV RNA into HIV DNA using an enzyme called reverse transcriptase. If HIV virus succeeds in it, then it can easily enter DNA into the nucleus of T helper cell and control it. As HIV fuses with the cell, it starts making long strands of messenger RNA protein and transport them toward the edge of the cell. This is then used for producing more HIV DNA. Now, fourth and last stage is the assembly, building and maturation. These strands now contain HIV genetic material. Using the genetic material, it is very easy to form new HIV particle, which takes place and then released into the T helper cells, and this spreads infection all over the body. Now, as we talked about, how does CRISPR help in curing the HIV? CRISPR is a tool, as we talked about, is a genome editing tool which can easily cut the particular sequence from the body and remove it as you wish, or insert the new sequence as you wish, and Cas9 really help it in it as acting as a scissor. So, CRISPR-Cas9 as a genome editing tool help in removing the HIV from the body. Now, what you can do is we can make a particular gRNA which can match the HIV DNA sequence and remove it from the body using the Cas9 enzyme. So, we will talk about the experiments conducted on it, as we have reviewed in the report the paper written by Chinese scientists like Visuan Jui, Rongyu Liu, Ryan Li Duff. The name of the paper was said CRISPR-Cas9 system inactivate latent HIV proviral DNA. So, how the test was really conducted? In the test, they first took the JLAT, Jurakat latency 10.6 cell, which contained the full length of HIV viral DNA, and they put a GFP, green fluorescent protein cell, which can monitor the viral gene expression. Now, to make the CRISPR-Cas9 identify the site, they need to identify the PAM sites, which helps in CRISPR identifying the site. The PAM site is the prototype adjacent motif, an immediate 3-letter nucleotide sequence after the gRNA sequence. So now after that, they identify the 80 PAM sites and identify the potential sites which can be used for CRISPR-Cas9 experiment. Now after this, they form 10 different gRNAs named from T1 to T10. So, of these 10 target sites we choose to make in the gRNA, 3 are in LTR, 5 in pole gene and 2 in the second exon of lat or reverse. So these RNAs are nucleotranscripted together with Cas9 plasmid DNA into J-late 10.6 with a Jurkat latency 10.6L. So the targeted viral DNA regions were also amplified by PCR and further examined by sequencing. Now, as we moved forward with the experiment, we conducted the experiment, we see that the experiment was successful, very much successful, but the most successful of these gRNAs was a T10, which succeeded in removing the 20 folds from the HIV DNA. As the least removal is by the T3, which is the 3-fold removal, or as you can see in the report, there is a diagram of score and CRISPR design on the page number 9. In the score in CRISPR design, the T10 was given the least, T10 was given the second least or the third least in the score in the CRISPR design, and T3 was given the much better score than the T10, but still the T10 was more successful than the T3. Now as we move ahead, we see that after the experiment was conducted, we see that there is a cellular transcription machinery contains an inaccessibility to LTR promoter in J-late 10.6L. So to stimulate it, to make it more accessible, we done a 16-hour prior treatment with TNF alpha. So we thought it will make it more accessible and it will be more helpful to the CRISPR-Cas9. But in contradiction, it does not affected or modified HIV DNA, making it more susceptible for the CRISPR-Cas9. Does not had any effect. So that experiment was as it is that the TNF alpha or making the susceptible HIV DNA does not help CRISPR anything alone. And as we see, if we did the experiment without using the Cas9, with the gRNA alone, there was no difference in the experiment, as it was of course, because Cas9 is the Caesar which is used for the CRISPR tool. So now we started making combinations of the gRNA, seeing it that how much potential the combinations have more than the single gRNA. So we first make the combination of T1 T6 T9, T2 T4 T10 and T3 T6 T9. It also succeeded to some extent. Both of these combinations removed more than 24-fold monitored by GFP positive cells. So we see that in summary, we can see that in order to measure the effect of gRNA Cas9 induced DNA, mutations on the function of integrated HIV1 DNA, we first transfected the J-late 10.6L with gRNA Cas9 DNA, followed by TNF alpha treatment to induce viral gene expression. Then viral gene expression is measured by using a method where GFP positive cells are scored by flow cytometry. So the observations we made in this report was that the T10 was the most successful of the gRNAs, which reduced 20-fold from the HIV DNA, and T3 was the least successful, which reduced only 3-fold in the DNA. And the gRNA alone did not have any effect or exerted no effect on the GFP expression without the help of the Cas9, as it is usual. Cas9 is necessary for the CRISPR tool, or Cas9 is necessary for the CRISPR to work, we can say. So in both insertion and deletion, gRNA T4, T8 and T10 cause the most type of deletions and insertion, whereas gRNA 3 led to only 3-fold deletion. But nowadays, as we can see, the more and more experiments are conducted as CRISPR opened a new gate or a new path to many of the applications or many of the new technologies or many of the new cures for the medical industry. So as we work ahead, we see that the more and more studies are conducted, but in the recent result, as we can see, it was seen that HIV does beat the CRISPR in the first round. As there is a statement written by Dr. Chen Liang, which we read in an article. It's like the leftover HIV puts on a fake mustache disguise before going back to work, revising your immune system. So what does it mean? It means that HIV has got a brain of its own, which is more intelligent than the CRISPR. If CRISPR cuts the HIV DNA and if there is any leftover HIV DNA, it does combine with the body in such a way that it becomes a part of the body, making it difficult for the CRISPR-Cas9 to identify that particular HIV DNA to remove. Now, as the CRISPR-Cas9 gets out of the system or the complex gets out of the system, HIV DNA regenerates its process, redo the whole process and starts infecting the body again, making it again infected with the HIV virus. So we does lose hope in that, but there is still no more concrete conclusions made that CRISPR cannot help. CRISPR did help a lot in that HIV cure, so there is still hope, and it helped us in it. Did evolutionize our way of looking at the genetic diseases. Now in the conclusion of the presentation, I can only say that there's a lot more work done, particularly in the field of accuracy. how do we make it more accurate for the genome sequence which we are targeting, so that does not affect the off target sites. So it is more than just, as I've written, it is more than just putting out human cells in a Petri dish. So the scope of the idea, or the improvement idea we gave in the report, which can be that you can either target multiple sites together. See, if you target one site, HIV will regenerate and will going to infect the whole body again. But if you start targeting the multiple targets with one bullet, like see, if CRISPR targets more than 10 or 100 sites together in the HIV part, HIV does not get a chance to regenerate again and again. You will again be targeting the HIV sequence again and again, so there is no need to target single sites, which will give time HIV to regenerate again and again in the body. Or the other method, as I have written, that HIV virus particularly targets the T helper cell known as CD4 plus cell in the white blood cell, which are part of the immune system, which plays a very important part in the immune system. So if HIV has a design of itself which helps them to target specifically CD4 plus cell, so why not hijack the HIV itself? If you hijack the HIV itself to make it a more deliver method, make it uses it as delivery method, then it itself is a box which contains the cure for the HIV. If you use the HIV as a delivery method and make it more and put it in the body with the genome sequence to target, then HIV will identify itself. HIV will make no mistake, as HIV itself knows its sequence, which is very perfect according to the HIV DNA, because it will perfectly match with the DNA we are targeting. So HIV itself can act as a delivery method. There are other delivery methods too, but I say, isn't it great to fight HIV with HIV? As I written the references, we have reached the article from the medical news today. There was also an article written on the human immunodefensive virus infection. So there are many others, and the paper we have read is, I have already told the name in the presentation before. The paper we have read is CRISPR-Cas9 system inactivates latent HIV-1 proviral DNA. I would like to thank my instructor and the TA. I have learned a lot in this course about the CRISPR, and I really enjoy the topic, the CRISPR-Cas9 actually, because it really is a great application to be seen in the future, or to create many paths for the different diseases, as it helps us in the most interesting part which I find of the CRISPR-Cas9 is the transgenetic mutation. Thank you. I would like to finish the presentation. Thank you.